Morning, everyone. Today I'll be covering using pooled CRISPR screening for target identification, and then following that up with an arrayed CRISPR screen to validate candidate hits. And as Matt said, I'll be using this project called Project ENCORE as a case study for a real life example of using these approaches. So the outline of what I'm going to go through today is first, I thought I'd cover the different strengths that are offered by pooled versus arrayed CRISPR screening and the sort of approaches you might want to use them for. I'll then move on to talking about designing an arrayed validation screen once you've already performed a pooled CRISPR screen, and talk through some of the options that are available to you when you design this. And then finally, I'll walk you through Project ENCORE to showcase this strategy for target discovery. So a key distinction, which is within the name between pooled and arrayed CRISPR screens, is the format in which you mediate the genetic perturbation. So for pooled CRISPR screening, you're delivering a whole guide library to a bulk population of cells. Typically, you'll then introduce some form of challenge, whether that be introducing a drug or leaving the cells to grow for a particular period of time. But what you need to do at the end of that experiment is extract and sequence the genomic DNA there so that you understand which guides have been depleted or enriched over time. So in contrast, in an arrayed screen, the perturbation is split by reaction compartment, and that's typically a 96 or a 384 well plate. So you know before you do the experiment which wells have each guide present in them, and therefore your readout doesn't require sequencing. This means this format is amenable to expanding into higher resolution assays because you don't have to map back which guide you've introduced. To note at this stage, I think currently the line between these formats is starting to blur because we've got new technologies available, such as Perturb-seq, where you introduce a knockout and then perform single cell sequencing. And we also have optical pooled screening, but these two approaches are still very much used today for discovery. I've taken this image from this paper here, which is in Nature Reviews Methods Primers, and I highly recommend it if you want to do some further reading on the technologies that are available in this space. So a typical approach when you're looking for novel genes that are involved in a biological process that you're interested in is to use a pooled screen to identify candidate hits. And generally at this stage, you want to cast a wide net and capture the strongest phenotypes possible. Then once you've got your list of candidate genes, you can then move into an arrayed format to validate those effects. And again, that's where the separation in a microwell plate allows for higher resolution phenotypic assessment. And the split between these approaches is down to the difference in core components that make up these screening types. And I'll just go through these now to make that clearer. So the first is in the scale of targets that you can assess within one of these screens. So for pooled screens, typically you'll be looking at thousands, if not whole genome scale, in terms of your library of guides. Arrayed screens, because they are more complex in their format, you typically want to have a more focused library. So it can be between 10 to 100 candidates. Because of this difference in scale, the cell number required for each screen translates directly. So for pooled screens, you will need a high cell number to get sufficient coverage of that large library. Whereas for arrayed screens, because you know a priori which guide you've introduced in each well, you don't need as many cells for the process. As I have mentioned, the readouts between the two differ in their resolution. So for pooled screens, generally these are sequencing based and you're looking at strong phenotypes such as viability, loss or gain. These can include some form of flow cytometry enrichment of your desired population of cells. But the benefit of arrayed screens is that you can introduce high content measures of different phenotypes. So this can be imaging, transcriptomics, proteomics. And one key advantage to this is that all of these assay types can actually be conducted if you set it up right within the same plate. So you can get a rich amount of information from a single plate. The equipment required for the two different types is therefore different in that pooled screens, if you've got a cell model in place for your question and you're able to culture that and introduce guides into the system and can submit your samples for sequencing, then these are more feasible. Arrayed screens benefit from having high throughput instruments such as liquid handlers, plate readers, and microscopes. That's not to say you have to have these instruments available to you. It's just that you can often scale up in the number of things you assess if you do have them. Moving on to the data analysis, this is a generalization and pooled screens are certainly becoming more and more complex. So this isn't always the case, but there are established pipelines for processing pooled CRISPR screening data. Whereas with arrayed screens, depending on how bespoke your phenotypic assay is, you may require a bespoke data analysis pipeline to be generated for its analysis. Therefore, the kind of cost and complexity on a per target basis is generally lower if you think of the scale that you're performing for a pooled screen, whereas arrayed screens are higher in cost for both the instrumentation that can be needed and also the labor required to optimize those conditions for screening. What this all boils down to then is that pooled screens are very suited for target ID. So when you don't know which genes are involved in a process, these are really beneficial to identify candidate hits. And then arrayed screens are used once you've generated that list of candidates and you can introduce more specific phenotypic assays. And a key element that I haven't touched on yet that bridges these two screening approaches is how do you pick which candidates that emerge from your target ID screen to take forward for validation. And I will show an example of how we did this for Project ENCORE later on. But just to say, the process of this is not it's not an easy one. You can often be needing to filter your hit list down from thousands of potential candidate genes down to less than 100. The first two steps of this process will be pretty similar for most pooled CRISPR screens. So there'll be some element of read count processing where you normalize your counts and filter out low count guides to reduce the noise in your screening data set. And then there will be some form of statistical analysis. So you'll calculate log fold change in guide abundance or guide depletion, a associated p-value, so statistical significance for that, and then some control for your false discovery rate because you're assessing so many guides at the same time. The next two steps in reducing your hit list down to something feasible for arrayed screening is firstly thresholding, where you'll often need to apply multiple thresholds for effect size that you see in your pooled screen, some threshold on significance, and there can be another threshold on consistency or reproducibility if you've screened multiple models or performed multiple replicates. And then the second step is considering the biological context. So it could be that you want to prioritize hits that are known to belong to relevant pathways or gene families that you're interested in working on. And it could be the other end of the spectrum, which is that you're really interested in novelty. So things that have been previously uncharacterized in the context of what you're investigating. But these two steps will be highly dependent on what biological question you're asking and what your validation strategy is. And so how many targets you can take forward. So before I move on to how you go about designing an arrayed validation screen, I thought I would just briefly emphasize the importance of a validation screen in the context of a drug discovery pipeline. And this comes from the work I was doing in the Open Targets Validation Lab, where this was the focus of what we were doing. This image here shows a problem that's faced by the pharmaceutical development industry, and that's the high attrition rate of targets once they reach clinical trials. And this is primarily driven by a lack of safety or efficacy shown for targets once they reach this stage. And it's clearly crucial that we reduce this attrition rate and ensure that everyone is spending their efforts on the most robust targets that are likely to make it to drug approval. So the aim of what we were doing in Open Targets and what the validation lab aimed to do was to address this issue by generating additional genetic and experimental evidence to ensure that the best targets are brought forward. This is showing you the importance of drug discovery pipeline, but I also think this is key for purely academically focused research to address the reproducibility crisis and ensure that any published biological findings have been really robustly scrutinized and validated in their system. So I'm now going to move on to talk about how to design a validation screen once you've performed an initial target ID screen. Typically, if you've performed a whole genome target ID screen, you'll get an output that looks something like this, where you've got response on the x-axis, that can be log-fold change in guide abundance, and a p-value associated with that response. And what you'll want to do, as I said, is apply a threshold, pick targets that fall above or below that threshold, depending on the direction you're looking at, or you can perform a ranking step in terms of the effect size of your targets. And as I said, I'll talk more about a specific case study of that when I talk about Project ENCORE. When you then move on to designing your validation screen, there's a key decision to be made on how equivalent you want that screen to be to your initial pooled screen. So what I've got here is a sliding scale where if we consider up here the equivalence to pooled screen to be a technical replicate, so you perform the exact same whole genome screen. And down the other end of the spectrum is, say, moving into a knockout mouse model with a very limited set of targets. As you move away from your initial pooled screen, clearly your assay complexity is going to increase if you're introducing increased phenotypic resolution. Because of that increased complexity, the number of targets that you can assess at the same time will reduce. But the trade-off of this is that you get increased phenotypic resolution. And it's to be expected that the more orthogonal you go relative to your initial target ID screen, your validation rate is going to drop. But it doesn't sound it, but it is a good thing, because one of the purposes of doing these validation screens is to weed out false positives and ensure that what you've seen is a genuine reproducible effect. So if we look into where we could place ourselves if we're designing a target ID screen, as I said, if we sat up here and performed a technical replicate of the pooled screen, this is verification of your pooled screening data set. That is something that is required to filter out noise that's inherent to the system. But by doing this, you're not going to be able to filter out any false positives or false negatives that are specific to that screening system. Going to the other extreme is something that can be thought of as picking the hero gene. So you just take the #1 performing hit within your CRISPR screen. This can work. It's not a criticism of this approach. It depends completely on what your strategy for the project is. And if you found the #1 hit and you're confident in it, then this can be the approach that you need to take. But you do lose out on targets that can sit in lower quartiles of your data set. And you're susceptible to focusing your efforts on something that may not validate later on. So it is beneficial to validate multiple hits if possible. So as I said, when I was working in the validation lab, we tried to position ourselves somewhere here on that scale of equivalence to pooled screen. And what that meant is we were trying to validate targets at scale, but providing additional mechanistic insights. So increasing the phenotypic resolution, but still maintaining a sufficient target number that we weren't focusing all our efforts on only a few. And this had two benefits. So obviously we were able to validate multiple novel targets, add mechanistic insight, and if we found targets that were validated, we could accelerate their selection as candidates for drug discovery. But the second benefit is that we got an idea of how the screen worked as a whole. So we could look into validation and invalidation rates by comparing the array to the pooled screening data set and start to begin to understand why some targets are failing validation. So when you come to designing a arrayed CRISPR validation screen, what options do you have when you design it, and where do you sit along this scale of equivalence to your pooled screen? The way I like to think about it is the three kind of pillars of a CRISPR screen, which are the model that you're going to use, the perturbation method that you'll use, and the assay that you'll read out the effect. When we think about the model, the generalization is that in your pooled CRISPR screen, you will have used some form of a cell line. That's not always the case, but that is the most amenable way to introduce a large guide library. Once you move away from your pooled screen, you can start to consider more complex cell systems. So moving from 2D to 3D, introducing rare cell types, and potentially looking at co-culture. And that is where the benefit of arrayed screening in being able to use lower cell numbers comes in. When you consider the perturbation, how you are going to perturb your target and also how you're going to deliver that system to your cells, generally, one of the kind of common methods to use in the pooled CRISPR screen is to perform lentiviral transduction. So you deliver the guide to the cell, and this can be using Cas9 expressing cells. When you move into the arrayed space, you may want to consider using different Cas variants. I shouldn't say this in the CRISPR course, but you can consider moving away from CRISPR altogether and considering drug inhibition or a different modality of targeting your gene of interest. And then finally, with the assay, as I said, for pooled screening, typically this uses guide dropout as a readout or guide enrichment, depending on which way you're looking at things. You can introduce much higher content phenotypic assays in the arrayed space. So looking at things like imaging, single cell RNA-seq, or proteomics. And this is moving away from looking at the DNA level and starting to focus on the effects at the RNA and the protein level in the cell. So what we thought about in the validation lab when we were designing our arrayed screens was that we're tuning each of these elements depending on what biological question we were asking. And again, this will completely depend on the system that you're working in, but it does boil down to what is it that you want to see to increase your confidence in a target? What evidence do you need to be sure that the effect you're seeing is not just an artifact of the experimental setup? So to put all of what I've just talked about into context, I will now move on to talking about Project ENCORE. I'm showing you this slide just to show that this is a very large collaborative project in open targets that involves teams at the Sanger, at the EBI, and the pharmaceutical partners that work with open targets. The goal of Project ENCORE is to identify novel combination drug targets in tumors with an unmet clinical need. And the reasoning behind this is that a key challenge with cancer treatment is the emergence of resistance to monotherapies. And by taking a combinatorial approach, we can prevent or at least delay the emergence of this resistance. ENCORE focuses on two tumor types with unmet clinical need. These are KRAS mutant colorectal cancer and triple negative breast cancer. So I'll start by introducing the pooled target ID screen that was executed in the Garnet lab here at the Sanger. So to walk you through the approach, because we were looking at combinatorial effects, we were using a dual guide system, so targeting two genes simultaneously. And the library was designed to include 40 anchor genes, which are cancer driver genes, arrayed against 1,600 library genes, which are cancer-associated genes. And this brings the total number of dual guides to 65,000 different gene combinations. So these dual guides were delivered to Cas9 expressing cells using pooled lentiviral delivery. And you can see that a large number of Cas9 expressing cell lines were selected and screened, and these were chosen based on having a variety of different biomarker backgrounds so that we could assess the effect of genes in a specific mutant background context. And then the readout for these screens, so after delivery of the dual guide and growth for a set period of time, the genomic DNA was isolated, and then the abundance of guides was assessed by next generation sequencing. So to directly contrast that with the approach we took in the validation lab, we prioritized between 50 to 100 gene combinations from this initial list of 65,000, and I will talk about how that was done. These were guides against these combinations were delivered in 384-well arrayed format using liposuction. We selected cell lines from the initial pooled screen, again, to give a range of biomarker background, but also to make it feasible to do arrayed screening. It wouldn't really be feasible to do this in all of the cell lines that were initially screened. And then finally, one of the key elements of orthogonality was these multi-parametric readouts that were used to assess the effect of the gene combination. So we had three different readouts. These were endpoint viability, imaging of confluence over time, and also imaging of cell toxicity. And this allowed us to gain a bit more mechanistic understanding, building on what was shown in the pooled CRISPR screen. So to show you the actual validation workflow, as I mentioned, we selected hits from the primary screen, and our kind of biological question and what we wanted to focus on were combinations that were lethal and also exhibited synergy in the pooled CRISPR screen. And that just means, for those unfamiliar, that the combination of gene A and B has a greater effect than the additive effect of gene A plus gene B. So there's an additional effect happening once you knock out both together. As I said, we were also using cell lines from the primary screen. So at this stage, we're actually very equivalent on that sliding scale to the initial pooled screen. But where the orthogonality is brought in is by using different guide sequences, a different delivery system, and those three different functional readouts. So this together ensured that any combinations that we saw as validated through this pipeline would be of high biological relevance and high confidence, because they've shown a reproducible effect in two orthogonal screening systems. To just briefly illustrate the difference in the data output and the analysis of the two screens, with the primary screen, the readout, as I said, is log fold change in dropout of guide counts. And we selected targets that were showing a strong effect size, so a log fold change beyond a certain threshold that we considered to be lethal to the cells. And we also selected based on whether they showed a synergistic effect relative to targeting just one of the genes at a time. In the validation lab, because we had these three functional readouts, we could use a combination of the three while assessing a target to determine whether that combination is directly lethal to the cells, if it causes a late cytotoxicity effect, or it simply reduces cell proliferation rather than actually actively killing them. So this builds on the evidence that we see from the pooled screen and allows us to understand a bit more about how that combination is causing death of the tumor cells. Just to emphasize as well at this point that all this information comes from the same 384 well plate, and it's the arrayed format that allows for us to extract all that information. So I've said that I would cover how we narrowed down this list of 65,000 combinations to a validation list of 56. So for Project ENCORE, we were thresholding based on what our biological question was. So as I said, we wanted lethal and synergistic combinations. And for version 1 of the validation screens, we initially selected KRAS as a priority anchor gene. So this just means that we're selecting combinations that have KRAS plus a secondary target. And this decision was made based on biology. So these cells are KRAS dependent. And there are drugs available that target mutant forms of KRAS for treatment of colorectal cancer. So already we have narrowed down that list from 65,000 to 1,600. We then applied a threshold on lethality, and this was based on the log fold change that we saw for the guides within the pooled screen. We applied a exclusivity threshold, so we screened both KRAS mutant and KRAS wild type cells. We wanted the effect to be only seen in KRAS mutant lines, which would increase the specificity of any downstream drugs. We then also applied a synergy threshold. So by using a couple metrics that measured the degree of synergy, we could narrow down the list further. And then with the addition of some controls and some targets that were nominated by industry, this brought our total number of combinations to 56, which is more than viable to assess within a arrayed format. Just to note at this point, we had 5 positive and 5 negative controls within this validation library, and that's nearly 20% of what we're testing, which may seem like a lot, but I just wanted to emphasize that that's really important for understanding the performance of both the primary screen and the novel targets that you're looking at. So you can really make sure that your assay range is sufficient to look at novel hits. So next, I'm showing you what the data looks like when we ran those 56 targets through the arrayed validation pipeline. This plot illustrates the overall performance of the validation screen. I've got a representative KRAS mutant and a KRAS wild type cell line. Each dot on the plot represents the effect that a combination has on cell viability. So if we start by looking at the baseline, which is where we targeted two non-essential genes to control for the effect that we're introducing CRISPR perturbation, you can see, as expected, baseline effect is having no impact on viability, which is to be expected. We also included a highly penetrant positive control, which shows a strong impact on viability. And this just gave us an indication that we've got a good assay window within which we can assess novel combinations. I'm next highlighting a positive control that was included in the library. This is a paralog pair, MAP kinase 1 and 3, which illustrate quite nicely the concept of synthetic lethality between paralogs. So this is an effect we see with paralog pairs where the cell can tolerate loss of one of the pair, but as soon as you target both simultaneously, this induces cell death. And to show you that on the plot, what we can see is if we pair MAP kinase 1 guides with non-essential guides or MAP kinase 3 guides with non-essential guides, there's not much impact on cell viability. But as soon as we pair the MAP kinase 1 and 3 guides together, we see this dramatic loss in cell viability. Next, I'm showing you the impact of knocking out KRAS alone. It's paired with a non-essential control, but what you can see here is that as soon as you target KRAS in the KRAS mutant cells, there is a large decrease in viability. And this only has an impact in this cell line as they are dependent on KRAS for their growth. Then finally, the actual tested library, looking into the combinations of interest that were identified in the pooled screen. What we can see, if you just look at this line here, where we've got KRAS paired with the library, there is a degree of synthetic lethality that can already be seen more pronounced in the KRAS mutant lines. So we can see quite nicely that some of the combinations, at least that lie within this data set, are exhibiting synergy and will be validating what was seen in the pooled screen. I'm going to zoom into that data because that was the data set that we were most interested in and show you an example of a clinically relevant pair. This is KRAS EGFR. These are two genes that sit within the same signaling pathway, and actually drugs targeting each of these proteins have been approved as a combination therapy for the treatment of colorectal cancer. So what I'm showing you here is the target ID screen, the pooled screen on the left, and the target validation screen here on the right. I'll just draw your attention to the fact that we can see that KRAS and EGFR have a greater impact on viability than either gene alone, and this is specific to the KRAS mutant context. And what we see quite nicely is we can completely recapitulate that effect in our validation data set. So this is simply a proof of principle that we can capture combination targets that we expect to have effects based on the pooled screen and from published knowledge. I am now going to show you, I can't show you the ID of these genes, unfortunately, but I'm highlighting the fact that we could use this data then to zoom into combinations that are showing a synergistic effect, specifically in the KRAS mutant context within the pooled screen. And we are able to validate these 4 combinations within the arrayed validation screen. And again, this decrease in viability is specific to the KRAS mutant line, which is showing us that this effect is specific to that context. This is delving into what we kind of did next once we had identified these targets. And this is something that can be done when you're looking at genes that obviously sit within a signaling network. And looking at the interactions of those 4 genes that we identified in the validation and the initial pooled screen, we could see that all 4 of these genes sit within a network with EGFR and KRAS. And there's a total of 8 interactions between the 6 proteins. And a question we asked was, how likely is that to happen between the genes that we've got in our library? Because this is now biased towards things that we expect to have an effect with KRAS. So to investigate this, Heiko, who's at the EBI, performed an analysis where he selected 4 random genes from the combinations that were tested and counted the number of interactions and essentially iterated that 10,000 times. What you get out the other end is a p-value that suggests that the observation of this network in our validation data set is not a random effect. This is just to show you something that can be done once you've performed a pooled and arrayed screen and you've got high confidence hits. You can start to extract novel genetic networks based on this data and start to think about drug targets from the perspective of gene network interactions rather than just gene-gene. So I just wanted to touch on that to show you what we're kind of moving forward to working on now. The final point that I wanted to discuss is once you've performed your pooled CRISPR screen and your validation screen, how do you compare the data that you get out of the two? So with a pooled screen, your data output can be shown something like this. So you've got a distribution of log2 fold change against the number of guides showing that effect. Normally, this data will be centered around 0, as the majority of the genes that you test won't contribute to the process that's being investigated. That's to be expected. You'll have applied some sort of threshold to filter for the tail end of that distribution, and then take those forward for the validation screen. Sometimes, or commonly, the validation screening data will look something more like the right-hand side, and you'll have more of a skew towards your effect of interest. That's to be expected because you are picking those targets based on showing an effect in your pooled screening data set. And the degree of that skew will depend on how many targets you take forward for validation and also how equivalent your validation screen is to your pooled screen. So when you get to comparing the performance of a target between your pooled and your arrayed screen, obviously your primary focus and the thing you're going to be most excited about are targets that can be considered true positives. So these are targets that are hits based on your thresholding approach in both the pooled and the arrayed screen. You will also reveal what can be considered true negatives, so things that are non-hits in your primary screen and not hits in your arrayed validation screen. Just to say, I don't think I mentioned it before, I would advocate that when you design your validation screen, you do take forward some hits like this that aren't expected to have an effect. Because this is an important measure of the specificity of your initial pooled screen and shows you that you're capturing non-effects as well in your validation screen. You will also recover what can be considered false positive hits. And these are hits that were identified in the primary screen and invalidated in your validation screen. And then this is one of the key purposes of doing a validation screen is to weed these out, as you don't want to invest further effort into investigating them. The opposite side of that is you will get some false negatives. So things that weren't expected to be hits in the primary screen, but emerge as hits in your validation screen. I'll just say at this point, without further follow-up of these two types of targets, the false positives and false negatives, there's not really any way of knowing which of the screens is the right one. You would need to do much more follow-up to determine whether their classification as hits and non-hits is just an artifact of the thresholds that you're setting. Ultimately, there's no way to assign which is the correct screen. But as long as your negative and positive controls have performed as expected, you're going to be focusing mainly on the true positives, and you can build that confidence to invest your time and your resources into following these up. So I will end just with some take-home messages. Pooled and arrayed screens offer different strengths for different purposes. Pooled screens are very good for scale and for discovery, whereas arrayed screens offer a bit more phenotypic resolution and validation of the pooled screens, but they work best when used together. I hope I've convinced you that validation is essential to confirm the relevance of your hit to the biological system that you're working on, and also to reduce the time that you would otherwise waste on following up false positives. If you can introduce orthogonality at the validation stage, this is important to ensure that the findings you have aren't context dependent to the screen that you've performed and aren't just artifactual. And then finally, when you get to the data interpretation stage, this can require bespoke design for the validation screen based on your screening format. It's important to think about the assay range for your screens, as these will be different in the validation screen relative to your pooled screen. And this is really important to consider when you set your thresholds for classing a gene as a hit or not. Yep. And that's all for me. I can leave these messages up, and I'm happy to take any questions, any thoughts on what I've discussed today. And thank you very much for your attention.
This is more like is it technical? But in terms of the readout for the pool screen, because
Mm,
obviously it's at the end of it, it's an NGS dropout. So basically, what you're looking at is did the cells, well, which one survived, which one died, which
yeah.
one survived, right?
Yeah, exactly.
Is it possible, and this, to have alternative readouts, or is it at such large scale, basically viability is the only reasonable readout? I'm thinking about particularly a protein expression level.
Yeah, I mean, absolutely other options are available. I think for the scale for Project ENCORE, with the scale we were doing, that was kind of viability was the only really viable option. But yeah, absolutely, people will perform FACS-based screens if they look at protein expression on the surface or even internally. That can be possible that you enrich for things that have increased the expression of a target upon gene knockout. Yeah, it's not the only option.
Thank you. That's just something to my particular use case that I'm thinking about.
Yep.
So
Yeah. No,
perfect.
guide
Thank you so
dropout is
much.
not the only thing. Yeah.
Yeah, that's put a smile on my face. Thank you.
Eloise, I'll let you field all the rest of the questions that they're coming in.
Sure. I just see people's faces. So Bryony.
Yes, thank you so much. That was really interesting. So I have maybe just more of a basic kind of question. I'm wondering how you choose your cell line for your initial screen. So
Mm,
obviously we have a habit of doing these kind of things in things like hex cells because they're the easiest ones to do.
yeah.
Obviously you're then with your hits, you're kind of you're biasing it towards just this
Hmm.
normal kind of cell line when if you have things like obviously you validated in your KRAS mutants and your KRAS wild types, but
Yeah.
how do you kind of choose what cell line to do initially?
Yeah, I mean it's I don't have an easy answer to it, unfortunately, because it does depend completely on what you're looking at. I think with what we did, we've obviously used cell lines in the pooled screen and then cell lines again in the validation screen. Something that would be really nice would then be to move into tumor organoids and go into something much more specific. It depends. Again, a lot of my answer are going to be it depends because if you need to go whole genome, potentially you do need to use a cell line to get sufficient numbers. But if you've got a model that's more relevant to your question, that can be grown up to the scale that you need to cover your library, then that would be brilliant. But I would say, as long as you have in your mind that you will follow up with something more relevant, then it's okay to start with something that you can at least perform that screen to get an initial idea of which genes are involved in whatever you're focusing on. Yeah, it's not I can't give an absolute answer because it just does depend on what you're looking at, I think.
Yeah, thank you very much.
Luigi?
Hi, thank you for the beautiful talk.
Thank you.
Sorry for the probable silly questions, but you mentioned that you may want when you go from the pool to an array screening to like change the delivery methods of the
Mhm.
guide RNAs, right?
Mhm.
Why is that?
So it effectively it takes out any artifact that you're seeing that is due to the delivery method you're using. It wouldn't be as extreme as this, but let me use this as an example. So if you're using lentiviral delivery and your cells have a specific response to the fact that they are being infected by a virus, and that gives you that say kills the cell and it gives you a false positive effectively that guide has introduced a viability effect when actually it's just the cell couldn't tolerate the infection. So by switching to a different method, none of them are going to be perfect, they will all have their own artifact, but you can hopefully they're not going to show the same effect in both delivery system. So it's trying to just switch it out so that you remove those artifacts associated with the delivery method.
Okay, thank you. This can also work backward, right? You can also see like some effect in the array screening by using, for example, transfection that you don't see in the others that
Of
were
course.
result from artifacts anyway. Thank
It's
you.
always that we're modeling and no model is perfect and there's always going to be something wrong with it. So this is why we do it multiple times, right? Yeah.
Thank you. Thank you.
Yes, hi. Thank you so much for the amazing presentation. So my question is, regards of, I mean, the ENCORE seems to be very statistically sensitive. And I was thinking about a way to like optimize my experiment, because you've mentioned that when leveraging like a pooled screen readouts seems to be more cumbersome. And would it be useful to use more stringent criteria with my guide RNA? Because like selection, when I'm targeting these genes, like for example, looking for certain exons that seem to be, or certain variants that seem to be replicate in the studies, instead of going more exploratory, would this help me later on in dealing more easily with, just analyzing the data?
I'm not sure I completely followed so your question is on selecting the guides in your initial pooled screen.
Yeah. So when I come to the statistical analysis stage, would it be more helpful for me to actually reduce the guide count that I had from the start in targeting these genes in my pool screen, or would it be okay for me to go as exponential as possible? Would that at all affect my analysis strategy, or make it better or worse somehow,
I think
basically?
it sort of depends on, I think the general consensus is more guides. If I'm understanding your question correctly, let me know if I'm not, but I think more guides per gene is beneficial for your statistical analysis. But if these guides have already been tested and you know which out of the, say, a pool of 5 are working, then it's okay to just go with using, say, 2 of them if they've already been tested. But it depends if that effect is known. Not sure I've answered your question. If I haven't, put it in the chat and I can
Okay.
think about it
Thank
more
you.
and let you know. Yeah.
No, that was helpful, but I think I would put more clarification. I just want to make room for others also to ask. So
Okay.
thanks.
Thank you.
Yeah, thank you for your talk. That was super helpful and very interesting. One question I had, so when you choose the validation approach, as you said, you want to kind of have some variability there so that you make sure that it's really a robust effect that you see and it's not just biased based on the approach you did in the first screening. But did you also try different validation approaches and then you correlated it? And do you then always find, let's say, the strong hits irrelevant of which approach you test, or how did you look at that? Because that's something
It's a really good question. I think something we were actually really interested in is that we do get some hits that only come through one of the assays. But that's sort of informative in and of itself, in that we can say, okay, this is clearly something that stops the cells growing, but we don't see a strong effect in the endpoint viability assay, say. So I would say that obviously as a presenter, it seems like a wonderful validation pipeline that's all built. This was kind of built on gradually. So the first was to introduce that endpoint viability assay. And then you move on to, well, we've got these in 384 well format. We may as well image these because we've got a system that enables this, and then we can increase the resolution by adding a toxicity dye. So this was built over a period of many years to get to the stage that it was at. But yeah, we do see things that only validate in one of the assays and not the others.
Okay, thank you. Because that's always a bit of challenge, that like getting the hits, or let's say in the, let's say, screens that I've been involved in the past, getting the hits is not really the issue, but like then actually validating them. And
Yeah.
that sometimes has always been a challenge. I don't know what your recommendation would be exactly, like playing around, trying different validation approaches and then see which is the robust effect, or do you have some insights of what you would recommend?
I would say, I mean, if you've got an assay in mind, the best thing you can do is have a good set of positive controls to check what effect they have in that assay. And is that going to be a big enough dynamic range that you can then look at things you need to validate within that dynamic range? Yeah, that was, I think for what we were doing with Project ENCORE, for us, that was a set of paralog pairs, like I showed with the MAP kinase one. We had really like a long list of these, and it was about making sure we could detect that effect in the validation assay first before we even invest time in looking at, you know, our candidate hits. Yeah.
Yeah. Okay. Thank you.
I'll go to the first one. Zainab, is the data available from this project? It's not yet published. It is planning to be imminently. So it will be, but yeah, not yet. SKR, we'd love to get the protocols so we can optimize them for our cell lines and requirements. Again, I think that will come with publication. Ilaria asks, thanks, Eloise. Which cell model has been used for the ENCORE project? Were these cells naturally KRAS deficient, or did you knock out KRAS by CRISPR-Cas9 strategy and then add the other target knockouts on top? Yeah, good question. They are kind of natural mutants. They are either KRAS G12C or G12D mutants. And this comes from a biobank here at the Sanger of multiple different cell lines where we're able to select those with specific mutational contexts for the screening purpose. Sorry, I'm just reading through the other ones. Are pooled screens applicable to primary cells, or do they require uniformity in the population at baseline as is provided by a cell line? I think that my answer to this would be that they obviously benefit in the data noise by having a uniform population, but that doesn't mean that you can't control for that by applying a pooled screen to a primary cell line. I think the data analysis becomes more difficult, but that's not to say that it's not possible.
Would you suggest then, if you are, sorry, that was my question, if you are applying it to a primary cell line to reduce that noise, then you just need to scale up in terms of cell number to reduce the noise artifact potentially?
I mean, I guess, yeah, I don't know your context, but if you say if you've got primary cells and you know 20% of those are the population you're interested in, then you know already that you need 5 times the number of cells if you follow, because you're actually only targeting 20% of
yeah, I was
what I mean.
Yeah, I was thinking of like back sorting.
Okay.
From say peripheral blood, say for example B cells, or
Yeah.
maybe B cells is a bad example. I don't know, monocytes, whatever.
Yeah.
Or a subset of monocytes. But then you'll have a variety of different monocytes within the population at baseline.
Sure.
So which will have its own noise. But
Yeah.
that's maybe a more technical.
I can make a comment that some of Alexander Marston's team's work, they've done a lot of CRISPR screens in T cells, or from PBMCs, and that is so that's pooled screening at quite a large scale in T cells. And I think one thing you need to consider is donor variability probably in that setting. So if you were to repeat the screen with a different donor, you might get some different results given genetic differences. So that's one consideration which you probably won't need to have in the cell line setting, but certainly it will be possible. Some primary cells much more difficult to work with because they're less proliferative.
Yeah. Would you be kind enough to share if you Alexander Martins, did you say?
Martin, yeah, I'll send around some papers in a moment.
Thanks so much. That's really kind. Thank you. Sorry for the hijack.
Mona asked, in an arrayed screen using a 96-well plate, how many replicas do you recommend per guide RNA? Just to use ENCORE as an example, we actually had, for each target, we have 4 guides targeting it, and these were all within the same well. So we performed the transfection. We have the guides in arrayed format. We add the cells on top and then transfection reagent on top. So they're actually being transfected with 4 guides. And then in terms of replicates of that well, we had 3 wells with that same system. So there was always 3 technical data points for each guide.
Thank you.
I just had one last question, Eloise. So to validate a screen, obviously you have to have some of the same conditions as the pooled screen, but at the same time you want to validate in a slightly different setting to increase confidence. So in terms of guide RNAs and cell lines, how did you approach that problem? Did you have different guide sequences but partially overlapping? Were they completely overlapping, completely different? And same with the cell lines. And how much overlap did you see there?
So we used completely different guide sequences. So actually these were commercially available guides, and the initial pooled screen had one set of commercially available guides. We went with a completely different supplier, but ensured that, you know, the sequences don't overlap. They are different. The cell lines are exactly the same, and I think that's partly by design to ensure at least initially we are, because I suppose we were already narrowing down from 22 cell lines to 7. And we wanted that biomarker background that's present in each of those 7 cell lines to be the same, so we could actually compare the effect of mutational background across the 2. You could, like, it could have been done using a different approach. I suppose it could have been that you move into a different cell system. But that was the decision at the time, I suppose.
Yeah, I think sometimes for arrayed screens, you look for a completely different characteristic in the cell line versus a pooled screen. Transducibility, growth rate, things like that. So it's quite a challenge. I think there's one more question, and then we can call it at that. In the chat there.
Pooled activation screen would be useful or applicable to cancer drug resistance study. Yeah, I would say absolutely is the simple answer. I'm sure I imagine probably people are doing this. Yeah, that it would be completely applicable.